fellow at the London Institute for Policy Analysis, and I was called into the White House in 2020 to promote and get the concept of early drug treatment going, which was part of the National Pandemic Plan. So these were daily fights with Anthony Fauci and Peter Navarro and myself. So it was quite a ride. Can I have the first slide? I think it's important. I was asked to talk about gain of function and biological warfare. First slide, please. Thank you. You need to understand that biological warfare is it's a serious science. There's thousands of man-hours of experiments on this. In fact, 40 years of experience. And what it's all based upon is getting an infectious aerosol in the 1 to 5 micron size range and getting it downwind over a target. Initially, it was for tactical, for battlefield use. And as we'll see very shortly, this expanded to strategic nuclear weapons capability. So particles above 5 microns, they get trapped in the sort of bronchioles, and you need more infectious agent. Up 15, 20 microns, trapped in the nasal mucosa. We've got very good defenses that are constantly flushing our airways with mucus and sending it to our stomach for destruction. The 1 to 5 micron particles will get down near the alveoli and diffuse in, and most importantly, they'll be retained. So minimal number of infectious agents needed if you can get it in this small size range, and it behaves as a gas. It can penetrate buildings, diffuse in, it follows diffusion. So the further downwind it goes, the less diffuse. But in the particle size range, if you breathe it for a few minutes, you'll still get an infectious dose. We'll show you a graph on this. Next slide. The open air testing, this is a slide from the 1950s. You can see the equivalent animal tests, guinea pigs, non-human primates, and humans, volunteers, Seventh-day Adventists that wanted to serve their country, but they didn't want to carry a weapon, and they volunteered for actual live agent tests. No one was ever hurt. The minute they ran a temperature, high-dose antibiotics would be given. And but what this allowed is like actual field data. That was developed into the first tactical BW weapons. You can now predict if your BW attack will work or not before you do it, based on aerosol, based on particle concentration and meteorological conditions, and the target downwind that you're looking for. Next slide. This is a 1950s live agent test using a harmless bacterial simulant. Navy minesweepers sailed 2 miles off the coast of San Francisco at night under ideal meteorological conditions, 2-mile onshore breeze, and it dispensed 156 gallons of this agent. The aerosol, primary aerosol settled out, the large particles, and the invisible 1 to 5 micron aerosol blew out over the city, reached as far as Oakland. Now, San Francisco at the time didn't have the tall skyscrapers, so this attack would not work. And this should have remained classified. Leroy Fothergill declassified it out of Fort Detrick in the 1950s. He was in charge of what gets classified and what doesn't. But if it had been actual agent, 700,000 people would have died. So this started a tremendous, now this is a strategic problem. Nuclear weapons equivalent effects. Next slide. Of the 1,419 microorganisms that we know of that can infect humans, including fungi, viruses, bacteria, rickettsia, there's only 14 that are designated as tier 1 pathogens. Only 14 that can survive for a prolonged period of time in an aerosol, free aerosol without a lot of organic material around it, and withstand the osmotic effects of being in the air and the oxidation effects. And very few of them can withstand ultraviolet light, so these attacks are mostly for nighttime. But these programs, and they still exist, some of them, the U.S. shut theirs off in, I think it was 69. But we know that a number of countries have ongoing biological warfare programs, and there's a constant search to expand this Tier 1 pathogen list. This is where genetic manipulation can come into play. Next slide. So when we're talking gain-of-function experiments, it equates to any experiment that may increase a pathogen's growth rate, that can enhance symptom severity and tissue damage. Most viruses are very species specific. They and their host evolved together. When something gets ready to jump species, it's usually naturally in a threatened ecosystem, but you can modify the species with there's some techniques so that it will cross. Reductions in cell-mediated immunity. This is your actual defense, not antibodies. Cell-mediated immunity, cytotoxic T cells, are the bulldozers and the tanks that come in and destroy the factories, the cellular factories that are making the agent. And that keeps you alive long enough for antibodies to develop, non-specific immunoglobulin M, and then IgG, which is like snipers. So the tank runs over the infected cell, or the cell decides to kill itself through apoptosis. And any remaining free viral particles are taken out and controlled by the antibodies. So yeah, it's a two-step process, but it's cell-mediated immunity that's important. So any resistance to drugs or vaccines, antibiotics, or even diagnostics. Any increased risk that any GOF experiment could inadvertently contribute to pandemic or widespread illness. We'll talk about that in a second. COVID was not properly regulated, although it has very low death rate. Its infectiousness, well, you can see the havoc that it caused. So there's some rules that need to be urgently changed. Next slide. Mother Nature is the greatest practitioner of gain-of-function experiments through viral spillover events, like from bats into humans. If you get a high dose enough of virus, it doesn't matter if you're vaccinated or not. The sheer high dose will override that. And we know that for smallpox. Massive exposure, you're going to die. Well, you can be vaccinated all you want. If without drug treatment, specific drug treatment, the vaccine's not enough because of the time it takes even a secondary immune response to ramp up. The virus has you before then. So spillover events, these are usually the virus hasn't fully adapted to humans, and there's low risk of human-to-human transmission. Viral trafficking, because these viruses are being put into the environment, and we're not the only ones in the environment. The rodents pick it up, transmit it. This is very prominent in threatened high biodiversity areas where your animal population's drawing. Those viruses don't disappear, they jump hosts and find new hosts. And now this thing has passed through. It's a lot better at operating in a mammal, and this is an indication of an impending jump into humans. Borneo is a high-risk area that we're hoping to go over and study in a bit. And by this time, now you can have its efficient human-to-human transmission through aerosol droplets and the possibilities of a pandemic, depending on how it's managed. Next slide, please. So we have gain of function, but there's also loss of function experiments. These are attenuated organisms. And Louis Pasteur did this with the rabies vaccine, with chicken cholera, anthrax, and through serial passage, which is still done today, these things. These have led to some landmark scientific breakthroughs. Scientists are still using these old methods from the 1800s. And the problem is the gain of function and loss of function, it's dual-use technology. They're intertwined. And how do you control gain of function without overly restricting loss of function? And these are, can I have the next slide? So between 2000 and mid-2022, over 7,000 alterations in function papers were published. This was to actually learn what a gene does, and this can be in cancer research as well. It's not just infectious disease. Study a pathogen in an animal model of a human infection. One of my colleagues 20 years ago was able to change the Ebola virus from human, the Zaire strain, into a mouse-adapted Ebola, which is a lot more convenient to study new drug therapies and this type of thing. And, you know, you don't have to use the non-human primates. Or to develop a new vaccine strain. At the moment, the United States has the most published papers, 57% of change of function papers, followed by China at 21%, and the others altogether about 25%. So developing a new vaccination strain, these are usually loss of function studies. Next slide. Of the 1,419 different human infections, we only have 34 vaccines. That's after 150 years of research. So you can't make a vaccine for everything. And this is one reason why there's such a prolonged testing period. You know, every year we have a drug come on the market and it's pulled after a year or two, or should have been pulled. Because when you start giving anything to millions of people, the variation between people, you're going to start seeing things. So you have to be very cautious with this. That's the list to the your right of the vaccines. Not all of them are very good. Or they tried using subunit vaccines, which is a very bad idea for an RNA virus that's constantly mutating. By the time you get your vaccine out against a single antigen, the thing's already mutated naturally, and you'll never catch up. You cannot vaccinate your way out of a rapidly mutating RNA viral pandemic. It's impossible. Next slide, please. This is the most common method. It's serial passage, and you take a poor mouse and you injected it with a virus, and you wait till it has signs of disease like a hunched back, ruffled fur, weight loss, and you take a sample from it and put it into another mouse. And this goes on maybe 38 generations. But very slowly, we can use cell culture too. That's how the Ebola one was done. You use human cells. And eventually it changes species to where the Zaire didn't even like bat cells anymore. By the way, when the coronavirus came out, it didn't like bat cells anymore either. It had a very low affinity. What we're trying to say is this is a process in nature. This virus just doesn't suddenly appear perfectly adapted to infect man. That really doesn't happen. Next slide, please. Oh, we deleted that. Go ahead, next slide. Next slide. Next slide. One more punch. Okay, so the history, sort of short history of gain of function research. Back in the 50s, scientists realized this concept of serial passage of a pathogen, that most often you would weaken it. Occasionally you would make it more virulent, but most often you would weaken it, and these are accumulated mutations. It's a process of something called Mueller's ratchet, where eventually the mutations aren't harmful, they're not helpful, but it's excess genetic baggage, and the virus loses its overall fitness. That's how Pasteur would passage things in dog spinal cords until it weakened, rabbit spinal cords. And that was the first sort of rabies vaccine. Next punch of the button. By the 1980s, the former USSR had weaponized full-length multi-drug antibiotic resistance into their A4 strain of anthrax. This was under the Biopreparat program. They became frightened of the U.S. Star Wars program and made significant advances in offensive biological weapons during that time. Hit the button again, please. The real worry in 1998, civilian scientists in England constructed an altered vaccinia virus, making it lethal. This is the normal strain we use for your smallpox inoculation. Sorry, I think it's this arm, I can't remember. Next button. And then in 2001, Australian researchers inserted the interleukin-4 gene into mouse pox, ectromelia. And this is an innocuous disease in mice. It gives them little blisters on their foot pads for a week or so, but they don't die. And this recombinant virus not only killed the whole colony, but it was resistant to cidofovir from Gilead, which we were stockpiling in case we were attacked with smallpox. So this is quite shocking stuff. Next slide. So 2014, we declared a moratorium on gain-of-function research. Let's stop and have an honest discussion about this. Do we need it? How can we do it without, interrupting cancer research and stuff? And one more button press. Moratorium end in 2017. Already? Okay, next slide. We got to go quick. So, gain of function, you can't hide it. Next slide. The government recommendations, NIH has made it. Office of Science and Technology have made recommendations. They're still not adequate. Next slide. There's been 3, 4, 5 reports from the National Science Advisory Board for Biosecurity. Again, they're not adequate. Next slide. Only 15 agents are covered. There needs more. Next slide. The real worry is the synthetic organisms now. Craig Fenter has recreated a mycoplasma organism from basic free nucleotides that he put into a shell of a mycoplasma. Next slide. Problem is dual-use facilities. This is Al Hakam in Iraq. Was completely missed during the first Gulf War and the second Gulf War. It's probably was one of the finest BW agent productions on the planet. Completely missed. Next slide. Vector, Novosibirsk making smallpox. Next slide. Accidents have occurred. Next slide. This is Sverdlovsk, an inadvertent anthrax release. Crude preparation went down 40 kilometers. Next slide. The main threat for gain of function remains civilian research under BSL-3 conditions. We've gotten a lot better at it. In '76, there were 4,079 laboratory-associated infections. From 2004 to 2010, there's only been 11. Next slide. Next slide. The way to get rid of this is with Class 2 biological safety cabinets that did the most, and proper protective gear, including full face covers, Raycal positive pressure. You tuck the Raycal hood into your Tyvek, and it puffs your suit out so you're under positive pressure all the time. These are the most common viral families that are being researched. We have 20,000 existing pharmaceutical compounds that probably have dual-use antiviral, some of them for certain. We just found that spironolactone, the diuretic, will completely block Epstein-Barr virus replication. So these things are hiding in there, and we need to start looking at these. Last slide. There's the example of the hood and working in a Class 2 biosafety cabinet. Last slide. Okay. The pandemic plan developed by the United States was from the top down. Last slide, didn't work. You need a new key from the bottom up. Last slide. And this is called a modular medical system, and it's based on local communities and the local hospital. And if a hospital can manage its own local epidemic, the nation can manage a pandemic. Okay, that's it.